Okay, well, thank you very much for the introduction. Thank you for having me. It's a great pleasure to be here. But why did you have to put me after Fang Zhang, Carolyn Bertozzi, and a charismatic chancellor? I think I would rather be interviewed by Lowell Wood and Tom Weaver than do this, but I'll try to do my best. So today I'm going to tell you, I'm going to try to keep it brief and tell you a short story. And what I'd like to do is actually pose a challenge that is still unmet, in my opinion, and that I would love to get Hertz Fellows thinking about. And then I'll show you kind of one approach that we've been taking to try to solve that problem. But I think there would be other ones that would be fun to brainstorm about as well. And that is the challenge of how can we image and control the function of cells deep inside the body, in the human body, but even in small organisms like mice that we want to study in the lab. And the reason that challenge is important is because for basic biology, if we want to know how neurons work in our brain, or what microbes are doing in our gut, or how immune cells are finding and eliminating tumors inside our body, to really understand that, I think we need to study it within the context of intact organisms and tissues and all the other cell types that are present there. In addition, there's an entire exploding field of cell-based therapeutics that's emerging. So things like CAR-T immunotherapy for cancer, or engineered probiotics that now people are developing that you can eat that'll go in your gut, detect inflammation, and release anti-inflammatory compounds, et cetera. And I think it's important when doctors administer these therapies into patients that we have good ways to check where are they? Did they get to the right place? Are they doing the right thing? And also to tell those cells what to do based on where they are inside the body. Okay, so that's the problem. Now, the reason this is a challenging problem is because if you think about the best technologies we have for imaging and controlling cells, they're all based on light. Okay, there are things like fluorescent proteins, which were in part developed by Roger Chen, when he was here at UCSD, or optogenetics that Fang Zhang contributed to during his PhD. These are wonderful tools that allow you to image and control what cells are doing because they're genetically encodable, and so we have very good ways to plug them into what cells are doing. But the problem is that light doesn't get very deep inside the body. Right? Photons get scattered within about a millimeter, and so it's hard to scale these techniques up to intact organisms, even relatively small ones like mice. So we need something else. And in fact, there are technologies that use other forms of energy. So instead of light, they use sound waves, or they use magnetic fields, and they underlie techniques like ultrasound and magnetic resonance that are ubiquitous in hospitals. But historically, these technologies don't show us anything about cells. They're showing us anatomy, where the bones are, where the muscles are. They might show us some blood flow and things like that, but they're not showing what cells are doing. And so that's the gap that we need to bridge. And the way we approach that is through biomolecular engineering, meaning that we find proteins and other genetically encodable materials that a cell can make that have the right physical properties to, instead of interacting with light, to interact with sound waves and magnetic fields. And the work in our lab over the last 10 years really breaks down along the lines of sound waves and magnetic fields and devising ways to use them to image and control cells. But today, I'm going to talk exclusively about sound waves, and I'm going to talk almost exclusively about imaging, but then I'll say a little bit about control at the end as well. Okay? So why do we love ultrasound? Well, it's a great combination of simple and powerful. It's simple in the sense that it's just based on sound waves that we transmit into a tissue that travel until they encounter an interface with something that has a different density or stiffness, like bone versus muscle. Some of that sound wave gets reflected back, and based on how long it took between emission and receiving the echo, we can tell how deep that interface is, and then we can scan left and right and generate an image like this one on the bottom left that you've all seen, and most of you were in at an early point in your life. And you can tell right away from this image that ultrasound can go deep, right? This is like a couple decimeters into the body. But what's harder to appreciate from these kind of images is that ultrasound actually is a very high performance technique. So in our lab, routinely, without any special tricks, we get spatial resolution, centimeters deep in tissue, of about 100 microns. Okay? So that's not quite at the level of a single cell, but it is at the level that's relevant for many aspects of biological organization in tissues, the brain, tumors, the liver, et cetera. It's also very fast. We can get sub-millisecond temporal resolution, so we can follow very fast dynamics with ultrasound. And there have been two things that have made ultrasound even more powerful in recent years. One is the development of super-resolution ultrasound, which is based on stochastic signal processes similar to the ones that underlie super-resolution optical microscopy. And so this is just an example from our collaborators in Paris, where the image you're looking at is a rat brain in a living rat, where the microvasculature there is resolved at better than 10 micron spatial resolution, and a living animal centimeters deep. So really amazing performance. And while being high performance, ultrasound is also very accessible. So today, for about $3,000, you can buy one of these ultrasound probes from Butterfly Systems, which actually was developed by a Hertz Fellow. And you're connected to your iPhone, and you're off and running doing ultrasound. So in principle, every biology lab in the world could be doing ultrasound to image cells, but they're not. And I think that's because, again, while we're really good at anatomy, we're not as good at connecting it to what cells are doing. And so our inspiration for making that connection comes from the history of fluorescent proteins, where there was an enormous leap in going from synthetic fluorophores to ones that are genetically encoded in GFP, when green fluorescent protein was identified in jellyfish and then transplanted into all the cells that we study in our labs. And so we want to do something similar for ultrasound. And our analog of synthetic dyes are these contrast agents called microbubbles, which, as the name suggests, are micron-sized bubbles of gas, typically stabilized by a lipid shell. And because these bubbles have a lower density and are more compressible than blood or tissue, they'll scatter ultrasound. Okay, and so if you inject these bubbles into the vasculature, that's how you get these beautiful images of blood vessels. But the problem is these are synthetic. They're thermodynamically unstable. The gas wants to dissolve. And so there's no way you're going to connect these to the function of specific cells. And so the question that we've been obsessed with for more than a decade now is there something in nature that's genetically encodable, like GFP, but has similar physical properties to a bubble? Okay, where can we find that? Because if we can't find something like that, then we hypothesize we'd be able to connect that to ultrasound. And luckily here, Mother Nature gave us a gift. And it came from organisms similar to the ones pictured here from the South Bay salt ponds near San Francisco, where the color in these bodies of water comes from photosynthetic microbes that had to solve an evolutionary problem of learning how to float. Because they use sunlight for photosynthesis, and they can only efficiently get that sunlight if they're near the top of the water. And so the way they evolved to do that is by forming these really beautiful intracellular self-assembled protein nanostructures called gas vesicle. Okay, intracellular, made inside the cell, self-assembling from protein components, nanostructures. This is an electron micrograph of one of these. You can see this gas vesicle that we purified from a cyanobacterium is a couple hundred nanometers in size. And what's special about this protein that makes it unlike any other protein that I know about is that it's a protein filled with air. Okay, so what you have is a 2 nanometer thick shell made of protein that encloses this couple hundred nanometer compartment that excludes water, but allows gas molecules to partition in and out, and thereby creates this little gas compartment. Okay? And when these cells want to float higher, they turn on the genetic program that encodes the formation of these gas vesicles to make more of them, and they float to the top in the water. And then when the cell wants to sink, they can break these down with proteases and sink back down. So really remarkable piece of evolution. And these gas vesicles, I bet even most biologists here hadn't previously heard about them, but they were originally discovered in 1895. So that citation I have there in German is from 1895. And then they were kind of a side curiosity of some biologists here and there, but the world largely forgot about them. So how did I find out about them? And this is where the Hertz Fellowship comes in. Okay, so I just want to briefly mention that. So when I was a postdoc at Berkeley, go Cal, I had no idea about gas vesicles, but I was interested in magnetosomes, which are these other beautiful nanostructures that are magnetic, formed inside cells. So I was reading a review article written by Hertz Fellow in my cohort, Michael Fischbach, about prokaryotic gene clusters, so these complex strings of genes that encode particular things like magnetosomes inside the cell. So I was reading this article, and he had a very brief section in there where he said a variety of species of archaea and bacteria control their buoyancy by forming gas-filled balloons in their cytoplasm. And I read that, and I was like, what the heck is he talking about? Because as an engineer, I know that it's very hard to make a stable nanoscale gas-filled compartment. And I said, oh, Fischbach must be wrong. He's a chemist. He doesn't know, sorry to Carolyn and others. He's a chemist. He doesn't know about surface tension. You know, it's totally fine, but this must be wrong. So I actually read those references, and I turned out that actually there's this thing that evolved called gas vesicles that make these little nanoscale structures. And that is what inspired me to then go and order these cyanobacteria from the Scottish algae collection and start doing experiments. And actually, just since I was walking down memory lane here, found a slide from a lab meeting that I gave when I was a Miller Fellow at Berkeley in 2012, where I showed the first data with ultrasound with gas vesicles. And this was fun because I ordered these bacteria. So those like little green dots you have there are these bacteria embedded in a big tub of agar. Berkeley is great at chemistry, terrible at ultrasound. So the only ultrasound I could find on the entire campus was the system from the 1980s that had a tape deck for the data. And then Patrick, my collaborator, and I went down to this basement and did the scanning, and I generously acknowledged him for XYZ positioning of the probe, as you can see in the slide. But the key thing is that in that image, in the ultrasound, we saw some contrast that was coming specifically from these cells. And so that's kind of what set us off on this journey. And so our more polished data that we published looks like this, where we purified some of these gas vesicles from cyanobacteria. We embed them in hydrogels, which are acoustically transparent. And then we have a concentration series of these gas vesicles. And the contrast that you see there is the ultrasound signal that's being scattered by them when we transmit it. And so that was exciting. And of course, one of the first things we said was, could we see it in a living animal? So we grabbed some mice, and we started injecting gas vesicles into their vasculature. It's under approved protocols. When you inject nanoscale particles, usually they get taken up by the liver. Okay, and so what we're going to look at in these images is that sliver that's outlined near the top is the liver, and it's initially dark. Okay, so just watch that. And as we start the video, we're doing the intravenous injection of gas vesicles, and over time, you're going to see that liver brightening up. Okay, and to our knowledge, this was the first time that a biomolecule could be seen non-invasively with ultrasound inside of a living, breathing creature. And so this launched a whole journey for us and created a new category of research that we call biomolecular ultrasound. And over the last 10 years or so, we've had a lot of fun just figuring out how far we can push this idea. And one really cool thing that I thought might appeal to this fellowship is that it's an incredibly interdisciplinary problem. Because on the one hand, we're dealing with these complex gene clusters and doing synthetic biology to transplant them around. On the other hand, we have to understand mechanics. Because the way these little nanostructures interact with sound waves depends on things like buckling that I had no idea about, but submarine people and airplane people study all the time. And then figuring out how to develop ultrasound techniques to interact with these structures as optimally as possible. So I'll just give you a couple of snippets of this in the next few slides. And so one of the biggest questions, and frankly areas of skepticism when we started, was whether we could ever take these genes and move them into mammalian cells. Because it's one thing to do that with GFP, which is just one gene encoding one protein and you get fluorescence. But it's another thing when you have this whole group of genes that have to work together. Because the gas vesicle, first of all, contains several different proteins within that nanostructure. And secondly, you have accessory proteins that are chaperones. They're like the construction cranes that are putting the structural proteins into the right place. And so they need to exist in the cell at the right stoichiometry and find each other and assemble. And so people, including reviewers of our manuscripts and proposals, were saying, this is cute, but you're not going to be able to do it in mammalian cells. But luckily at Caltech, and I'm sure it's true elsewhere as well, the harder the problem, the more the grad students want to do it. And so here I'm going to summarize 4 years of work by a brilliant grad student and collaborators with this little black arrow that's there, where we basically took all of those genes that encode gas vesicles and figured out how to string them together. So what you see on the right hand of the slide is genetic programs that use the latest tricks of synthetic biology, some of which are borrowed from viruses, some of which are borrowed from mammalian cells themselves, that allow us to program in these genes at the right stoichiometries and figure out that when we then take those genetic cassettes and put them into the genome of a mammalian cell, like this human cell on the bottom left, we can get gas vesicles formed inside. So that middle picture there is a thin section electron micrograph showing a bushel of gas vesicles forming inside the cell. And more importantly, that allows us now to image these cells with ultrasound. And so the data that you're seeing on the bottom there is live human cells that we've embedded into a hydrogel. And we're driving the expression of these gas vesicles with a promoter that's under our control with a chemical. So the more chemical we give, the more signal they should produce. And indeed, that's what we see in the data on the bottom as a function of the amount of this chemical and time after we administer it. So that's cool. So this shows us we can see live signals from gene expression in living cells with ultrasound. But of course, again, we want to do it in vivo. And as a first test, we created little tumors in mice. And there's a whole field of people studying oncology in mouse models. But it's very hard to see inside the tumors as they're growing. And so what we wanted to see is it's known that as these little tumors grow, they're better vascularized towards the perimeter of the tumor rather than the core. And so if you're asking the cells to produce a gene, they're going to have better metabolism towards the perimeter. So in principle, if we engineer these tumors to produce gas vesicles, we should see more signal towards the outside than we do in the middle. Okay? So we take our images, and indeed what we see is this. So in grayscale is the anatomy. So you see the skin at the top there, and the tumor is located right underneath the skin. And you see this oval kind of pattern of signal that's coming specifically from our gas vesicles, where it looks like it's mostly on the outside, and then there's dark core in the middle. And what we can do is then confirm this by taking out the tumor and sectioning it and looking at it with fluorescence. And you see this nice corresponding pattern of expression. And we saw this across multiple animals. The key is that the image on the right was taken after we terminated the experiment and cut the mouse into little pieces, and the experiment is over. Whereas the image on the left, the animal is alive and well, and we can continue to follow the dynamics of this process over time. So that was an important step. And just to show you the kind of stuff that we're doing with this now, just sticking with the cancer domain, is we're trying to study metastasis. And interestingly, even though most patients unfortunately die from metastasis rather than the primary tumor, most biologists are studying the primary tumor because it's much easier. It's easily accessible, you know where it is, whereas metastases are hard to see and follow, and so we're trying to do something about that. And so this is an example where we seed the mice with a tumor, and we're watching metastases form in the liver over time. And so over several weeks, in week 1, again, grayscale is the anatomy, and hopefully those of you who are close enough can see these little green dots that are in there. So that signal that's coming from tumors that are expressing gas vesicles that have metastasized into the liver. And at this point, they're still very tiny. This is like 100 micron or a couple hundred microns in size. We can already see them. And then in the same mouse, a week later, we see some of them growing, and we see new ones appearing. And a week after that, there's more of them. They're more extended. And so now what this lets us do is to follow the dynamics of these metastases and try to understand, well, first of all, the statistics of them. Are they all growing? Are some of them dying out? What's going on? And then now overlay on top of the different treatments and things that would help us study that. OK, so that's kind of a basic biology question. But then I also mentioned that we want to see what cell therapies are doing inside the body. And so one class of cell therapies that we're excited about are bacteria-based cell therapies. And in particular, there's been a lot of beautiful work, including from UCSD, from Jeff Hastie's group, on tumor-homing bacteria, where you inject them into the bloodstream. They can get into tumors and thrive within the interior of the tumor core, and there's been a lot of very nice work on that. But people have no way to see after they administer these cells whether they got to the right place. And so we engineered these therapeutic bacteria to produce gas vesicles, so we could see them with ultrasound. And I want to show you an image that I really like from the study. So this is a tumor in a mouse, and in red scale is an image of the vasculature within the tumor, and in green is signal that's coming from these bacteria using the gas vesicles. Okay, so we can see where they are. And what I like about this image is that it shows that these bacteria are actually infiltrating into the very core of the tumor, which is the opposite of what most drugs do. For most drugs, it's actually very hard to go far out of the vasculature. But these bacteria, because they're actively moving towards the hypoxic core and immunosuppressed core of the tumor, are able to get in there. And now we can follow that, and we can check that that's what's going on. And perhaps this could be useful in tracking a therapy in patients. So that's for imaging static signals, like where the cells are and gene expression. But can we image more dynamic signals? And we heard earlier in one of the Hertz Fellow talks about imaging calcium in the brain as a way to look at neural activity. And that's enabled by these fluorescent proteins based on GFP that have been engineered so that in the presence of calcium, their fluorescence signal changes. Okay, so you can see flashes of signal. And people have made sensors of enzymes based on similar principles as well. And so we want to do something like this, but for ultrasound, so we could do it non-invasively. And what allowed us to do that was some fundamental work connecting the protein composition of the gas vesicles to their mechanics. And in particular, there's this protein called Gvpc, or gas vesicle protein C, that is a rod-like alpha helical protein that sits on the surface of the gas vesicle shell and acts as a mechanical stiffener. So when the Gvpc is present, the gas vesicle is stiff. If you take the Gvpc away, the gas vesicle becomes more flexible. And then what happens is that when we transmit an ultrasound wave, which is a pressure wave trying to compress this and rarefact it, compress it and rarefact it, the gas vesicles that lack Gvpc and are more flexible are able to reversibly buckle and unbuckle, buckle and unbuckle under ultrasound. And what that does is produce a unique signature of sound waves that are coming out, this nonlinear scattering that's coming from the gas vesicle. And all you need to take away from this slide is if we don't have Gvpc, we see this nonlinear signal in blue there, whereas when Gvpc present, it's suppressed. Okay? And so now the way we take this mechanical insight and use it to make biosensors is by engineering that Gvpc protein. Okay, and the first time we did this, we made a sensor of enzymes, in particular proteases, which make a cut in other proteins. And just by introducing a recognition motif into that Gvpc protein that can be cut by a specific protease, we can make the signal dependent on that protease, because when it makes the cut, it takes that stiffening rod, cuts it in half, and it's no longer able to provide as much stiffness. Okay, and after a lot of tinkering and engineering, we showed that we can make sensors of several different kinds of proteases that are relevant. This is a viral protease, this is a human protease, this is a bacterial protease, and in all these cases, when we add it, we see the signal turning on. So that was a nice proof of concept, but I was a neuroscience major as an undergrad, and so I'm obsessed with the calcium imaging and seeing what's going on inside the brain. I'm going to probably skip this, but by the way, you can image the brain with ultrasound already based on blood flow. I'm not going to explain exactly how it works, but one cool thing I like about ultrasound is, again, unlike light, we can imagine translating it into human technology someday. And you would say, oh, ultrasound in the brain? The skull, what are you going to do about it? It's true, the skull is this hard material, but the nice thing is that we can actually replace a little piece of your skull. So we've developed with collaborators a polymer material that has protective mechanical properties similar to the skull that can replace a piece of skull, and we've actually put it into a human patient. And we didn't get any volunteers to do this, but there are patients that have traumatic brain injury where they have to remove a part of their skull, and then they replace it with reconstructive surgery. And we convinced our surgeon collaborator to make this implant, approved by all the authorities, whatever, that would have a polymeric window within this implant that when we put it onto the brain, okay, close everything up with the skull, then we can come in with an ultrasound transducer and image through this acoustic window. And in fact, it works. And so we have this patient who's a skateboarder, had a traumatic brain injury, loves guitar and video games, and this is us watching brain activity in his brain that's activated when he's strumming the guitar. And now we can see that very nicely with ultrasound. And this is just based on changes in blood flow that come from brain activity. In the long-term future, what I imagine is we'd be able to do this by looking at something like calcium and really see what's going on there. Okay, so the last couple minutes, I want to talk about the fact that ultrasound not only lets you image things, but also lets you control them. And in fact, everything I told you about so far is in just one regime of ultrasound where we send brief pings of sound that go in, bounce off, and come back. But if you have more extended pulses of ultrasound or different frequencies, then you can do different things. For example, if you have ultrasound with a gradient of the acoustic field strength, you can create force to move objects around. Or if you focus ultrasound on a spot and deposit energy, you can create heating and burn things or heat things up. And if you have bubbles present that you either create or already present that can expand and contract dramatically, you have what's called cavitation, which can produce very strong mechanical effects. And we've been interested in how we can harness each of these things for interacting with cells. And I just very briefly, as a teaser, tell you about a couple of them. Again, just staying with gas vesicles as the vehicle for doing that. So one thing ultrasound can do is push objects around. A very old experiment with sound waves from 1866 that's pictured here, where there's a sound wave that's sent into a capped tube. Some of the sound wave gets reflected back and creates a standing wave interference within the tube. And then you have particles within the tube that experience an acoustic radiation force that organizes them at the nodes of the standing wave. Okay? And this was actually used to measure the wavelengths of sound back then. You can easily do this at home. Just buy a speaker and a reflector from Amazon, and then you can suspend styrofoam balls in midair using this technique. Very easy experiment. You can invest in buying an array of ultrasound transducers and program with Arduinos to have a custom acoustic field pattern that will allow you to control these particles and have them dance around. So obviously, it would be really cool if we could do this with cells in a genetically selective way. And the thing that lets us do that is the fact that the same properties that make gas vesicles good at scattering sound waves also allow them to experience a strong force under ultrasound. And that's the difference in their density and compressibility relative to the surrounding medium. And in fact, if you calculate what's called the acoustic contrast factor, it's off the charts compared to other types of materials people have tried to manipulate. Again, because air is both less dense and much more compressible than water. And so we hypothesized that we'd be able to use ultrasound to manipulate gas vesicles and gas vesicle expressing cells. And I just want to show you a couple cool videos. So this is one where we have gas vesicle expressing cells that are inside of a microfluidic channel. We can see them with light. So those are the little grains that you see in there. So live cells. And we're going to apply ultrasound across this channel. So we have a node in the middle, and antinodes are maxima at the walls. And we expect these cells to experience a force towards the walls. Okay, so I'm going to turn on the ultrasound, so watch that channel. Whoop. And the cells migrate. And that only happens when these cells are expressing the gas vesicles, because otherwise they don't have enough of acoustic contrast to be moved by the ultrasound waves. We can do more fun things, like have multiple nodes and antinodes within the channel, have the cells organize, and then change the frequency. So it changes where the nodes are located, and we get the cells to dance around. We can create a focus and get the cells to organize at that focus. So initially, they're too dilute, but then as we keep the ultrasound turned on, they organize in this little ball, and then we can drag the ultrasound around and move the cells around with it. And then using acoustic holography, we can actually create an arbitrary 2D or 3D pattern of the ultrasound field. And for example, organize cells into the shape of a letter R, and then trap them into a hydrogel so they stay there in that shape. So that's cells that are trapped in the shape of a letter R. And we can see that both optically, just with the iPhone image, as well as with ultrasound. So what's better than moving cells around? Well, it's blowing them up. And so that's where cavitation comes in. And with cavitation, the idea is that you have several cycles of ultrasound that will interact with the bubble. Now, gas vesicles are not a bubble. Don't call them a bubble. They're a protein-shelled nanostructure filled with air. But we can turn them into a bubble. Okay? And the way we do that is by applying a strong enough ultrasound pressure that causes the gas vesicle to irreversibly collapse. Okay? So this is beyond buckling. It's just collapse. And when they collapse, the air that's inside of them gets released out. Initially, it's just a nanobubble. And usually these nanobubbles, because of the surface tension that Michael Fischbach was not aware of, they'll just dissolve within a millisecond. But if you apply sustained ultrasound, what happens is that the negative pressure part of the ultrasound cycle will cause that little nanobubble to expand and contract, expand and contract. And as that happens, neighboring nanobubbles experience an attraction force towards each other because of each other's acoustic field that they're generating and coalesce into a larger bubble that then can undergo this inertial cavitation and produce strong mechanical effects. And so we hypothesized this. Turned out to be true. If there are any ultrasound people in the audience, you can see that from this broadband emission shown there in orange. But for everybody else, we want to see it with our naked eyes. And so we borrowed a 5 million frame per second camera from our aeronautics department, right? Because ultrasound is happening at like a megahertz frequency, so we need something very fast. We suspended this expensive camera over a water tank where we're doing the ultrasound, and then we acquired these images. So what you see here are cancer cells to which we've targeted the gas vesicles by antibodies. Okay, so now they're attached to the cancer cell. And initially, we just see this texture of cells that's present there. The gas vesicles are too small to see. The ultrasound is already turned on, so watch the video. Okay? And after a few seconds of this video, which corresponds to a few microseconds, in reality, you start to see these black objects that form, and that's bubbles forming and cavitating. And again, that only happens when you have gas vesicles attached to those cells. And so what we've done here is converted our innocent little imaging agent into a bomb that we've attached to a cell to blow it up. And we can see that the cell is dead using staining. And then we can do this in vivo as well. I'll skip this, but you guys can see in the paper how we can use this to treat tumors. All right. So last thing, and this is David, I think, asked me to put in some structural stuff in here. So how are gas vesicles even possible? You might be wondering this too, right? Like, how did nature create a void inside a cell? How do you get a protein that is translated in an aqueous environment from mRNA and is able to somehow stay dry or become dry to form these kind of shells? And so one of the things that's been missing for a long time in the study of gas vesicles is what is their molecular structure? These are not proteins that are easy to crystallize for X-ray crystallography or anything like that. So we had to come up with variants of cryo-electron tomography to get at the structure, and finally we did. And so just zooming in a little bit on this molecular structure, this is the last data slide here. The gas vesicles turn out to be a corrugated cylinder, okay, with cones at the ends, where these corrugations are formed by a crystalline arrangement of a small protein subunit, only 7 kilodaltons in size, where the special feature of this protein subunit is that it has an extremely hydrophobic interior surface. Okay? So that's what prevents water. Even though water, presumably, like other gases, can come in as vapor into the structure, it can't condense into a liquid on the interior because of the high interfacial energy with the interior of this protein. And the exterior is hydrophilic. It's happy to hang out with water. But then the other question is, how does it get into that configuration? And so what's known is that the gas vesicles grow from the middle out. You have these little kernels, which are presumably just the very tips of the cones, and then that crystalline protein subunit gets added in between the cones in the way that causes the structure to grow from the middle out and expand, push aside the water, and create this essentially vacuum compartment that then gases go inside. And our former postdoc, who's now on the faculty at TU Delft, made this really nice illustration of how that works. So initially, in light color there, you just see the tips that are formed, and every colorful thing that's added is going to be these subunits that get added in as it grows. So watch the video. So you have them being added in the middle, and this protein nanostructure is growing from the middle out to form ultimately this cone with cylinders. So even though Michael is a chemist, he was right that these things do exist, and now we figured out molecularly how that works. So conclusions, ultrasound is pretty awesome, and it is just becoming possible to interface ultrasound with cells using genetic tools, and there are many possibilities and challenges. If anybody here is interested, I'm happy to talk to you about this whole area of research. This is the group that did the work that I showed, and I acknowledged people as we went along in the slides. This is from our beach party at Oceanside, not too far from here. And we're doing a lab retreat in Temecula. So I guess we have like a San Diego-ish party scene going on. And then the last slide I want to come back to is just this challenge of how can we talk to cells deep inside the body. And I showed you one way to do that, potentially, that we're still on the journey to figure out with ultrasound. But I would love, if somebody figures out how to do this with magnetic fields, let's say, right? Or other forms of radiation that can go deep. We've tried and failed, and I'm happy to tell you about the failures. But it would be awesome if other people work on that. So with that, thank you very much.